we're especially glad to have Sylvia Cremer with us today. I've wanted to get her here for some time, and it's been possible thanks to her also being the first person to do 3 different talks in 2 days, all of which are relevant to so many groups here at Arizona. I first learned about Sylvia when I was at the Institute for Advanced Study in Berlin in 2007, and everybody kept talking about the Sylvia Cremer. Do you know Sylvia Cremer? She does work about evolution and immunity in ants. And it turns out I started reading her work then, and it's profound, actually. Now, there is behavioral immunity, and her approach using deep evolutionary theory to try to apply those immunologic ideas to the behavior of ants, given their peculiar genetics and social organization, I think offers marvelous implications for other kinds of immunologic studies and human disease as well. I think there's tea time right afterwards, so anybody who wants to talk more with her will have an opportunity. I won't take more of your time now. We're so glad you're here, Sylvia. Welcome.
So I'm here to talk to you about the disease defenses in insect societies. So we are interested to see how diseases are spreading among societies of ants and what the ants can actually do to prevent this. As you know, sociality and disease often comes together, and this is the case for human societies and also for ant societies. And why do we have so many disease risk factors in these ant societies? So on one hand, you have an insect colony often consisting of very many highly related individuals. And that means that if you share the same genotype, you're often susceptible to the same diseases. So when a disease comes in that you're susceptible to, it can easily affect a high number of individuals in the colony. Okay. So also in these insect societies, you have overlapping generations. So the queen is laying their brood that is then reared basically by their own daughters. So you can have actually pathogen persistence over a long time across these generations in an insect colony. Moreover, there are very close social interactions that and they are very frequent among these individuals, which is increasing the risk of transmitting the disease from one individual to the next. And lastly, also these social insects, they shape their environment to make it a very stable temperature-controlled, humidity-controlled environment, which actually offers a very good microbial growth environment as well for the pathogens. And this is all what is maybe leading to an increased risk of disease in these insect societies. But actually, disease outbreaks are relatively rare. And this is because all these very sophisticated disease defense mechanisms that these social insects have evolved. So I like to see it as some kind of layered immunity. So you have very many different layers how an insect colony can prevent infection and disease. So this is a schematic ant here. So this individual has an own immune system. The insect immune system is very different from the vertebrate immune system, but it also actually functions relatively similar. So in the last 10 years, it was actually found that insects and crustaceans also, for example, have immune memory. One does not yet know what the mechanism is, but one knows that it's functioning very similar actually to the vertebrate immune system, but you don't have antibodies or lymphocytes, but you have some hemocytes and melanization processes going on. And each individual also has a behavioral immune component, something like disease or pathogen avoidance behaviors. And this can be summarized as the individual immunity of each colony member. But what makes these social insects very special in a way is that when they are interacting, all these individuals are interacting, a new layer of social immunity can emerge, and that comes from cooperative actions between these individuals. So they either do something together, like they change the temperature in the beehive, for example, or they do something towards one another where they really take care and perform sanitary care. And so this social immunity can then lead to a protection of the colony. And this protection of the colony can arise through these collective actions of the group members that are either joint efforts or directed towards one another. And the first step is always that they try to prevent infection after pathogen contamination. And so this is often achieved by sanitary care that is provided by healthy individuals towards contaminated individuals and can prevent them from falling sick. However, the second step is to try to prevent the transmission if some of the individuals become infected after all and are then infectious to the colony members. And the mechanisms that we find here are based a lot on behavior. So I'm a behavioral ecologist by training. So this was really what made me look into this. So we find a lot of nest hygiene and sanitary care. It also relies a lot on chemicals, so a lot of on antimicrobial compounds that are used, and on organization. So these mechanisms are behavior and chemistry, but also an aspect that I would like to talk to you mostly today is the organization of the colony. So these behavior and chemistry is something that can act equally well at the individual and at the colony level. However, organization is something that is really unique to these collective defenses. And so what I would like to put the focus on is studying these social interaction networks in the colony. So here you see a scheme of the colony. Typically you have one or sometimes several queens. She lays eggs that are then taken care of by workers to become larvae and larger larvae, and then they form pupae. And then the workers are emerging and take care of nest maintenance. All these individuals can interact, and there's also a lot of social interactions going on. But only recently it has become possible technically to really get a good picture at these interactions at the colony level. And this is due to some toolkit development where, for example, you can observe the social interactions of colonies by putting barcodes, gluing barcodes on each individual, and then make videos of these ants. So this is from a publication from Laurent Keller's group, with whom we collaborate now to take this into to not only look at social organization, but also to look at it in the disease context. And yeah, so so what the Keller group has actually found out using this technique is that the social interactions in a colony are not egalitarian. So it's not everybody interacts equally with everybody, but that you can really have a non-random social interactions. And that means that if you have the queen here, it's mostly taken care of by nurses. Then you have cleaners or individuals that take a lot of different tasks in the colony. And then lastly, you have the foragers that go out. And as you can see here, you have a lot of interactions within these tasks groups, but not so much between them. And interestingly, these different behaviors in ants, they are also depending on the age of the individuals. So when a new worker emerges, the first thing she does is to take care of the queen and the brood in the center of the colony, and only when she grows, when she ages, she will then take over these nest maintenance tasks more and more, and the foraging. So you can have a task-age correlation here. And what we can see is that the social interaction network is substructured into these communities of these age and task groups. And this was a very important finding for us because it showed us that the prerequisite for the organizational immunity hypothesis that we have been placing together with other people is fulfilled. And this means that in a colony, the idea is that you have a queen that is surrounded by the community of the nurses, and here you can see the brood items, and here, for example, the older foragers. And the idea is that if a disease comes in, for example, by contamination of a forager outside, it will spread more within the community of the foragers and has a harder time crossing these barriers to the other community of the queen. In addition, the other idea is that the ants do not only have to rely passively on their organization that they anyways have, but that they can also change and actively modulate their interaction network upon disease coming in. For example, they can break or interrupt some connections and, for example, quarantine individuals that are sick, or they can intensify sanitary care and things like that. So the idea of this organizational immunity hypothesis is that you have an inherent colony organization that is already limiting the possibility of disease spread, mostly from the outside towards the queen, and also that you can have network modulation upon infection to further limit disease spread, particularly to the most valuable individual in the colony, the reproductive queen. So basically, organizational immunity has both a prophylactic and an induced part to it. So this hypothesis is very well supported theoretically and also by studies that have used some non-pathogenic proxies. However, there's not really a lot of experimental work that is supporting that when you bring in the pathogen. So this is what we have been trying to do, and we have been trying to approach this in a dual approach. So on one hand, I call it the small scale approach. So what we do is that we use small subcolonies of ants, really something only like 10 individuals or so, and observe every type of behavioral interaction that we can see in a 1 hour or 3 hour interactions. So there we can study in detail the sanitary care actions and the behavioral sequences also. What does an ant do first? What does it do after it has had contact with an infectious individual, etc. And the other approach comes from the other side. It's the large-scale approach where we then look at complete colonies and use automated tracking, because if you have something like 150 to 200 colony members, it would be a nightmare basically to observe these videos by hand. And that means but also that you lose some detailed information. So what you do is that you infer the interactions only, but you get interaction networks, and you can do that over long periods of time, multiple days, whole colonies. So what we are interested to look at is what's the behavior, the inherent behavior before any infection, and what happens after exposure of the colony members. And what we have recently also started to develop tools for is to then measure directly the pathogen transmission. So how are the behavioral changes that we see in the hosts are really affecting directly how the pathogen moves from one individual to the next. We are also working together with people that perform epidemiological modeling to really see what would be the potential of disease transmission in these different types of interaction networks. And I would like to introduce the people that have been doing the work, both in my own team, so postdoc Line Ulvi and PhD student Barbara Casillas and Chris Pull. And we work together with people that are doing epidemiological modeling at the Helmholtz Center in Munich, and also with the Keller Group in Lausanne, and his postdoc Nathalie Stromayet. So the model system that we use is we use ant hosts and their fungal pathogens. And this pathogen is interesting, or gives us a lot of experimental opportunities, because we can rear it on agar plates in the laboratory, and then we can harvest these fungal spores. And what happens in nature is that if a spore or meets a host, basically, or it will attach to the host cuticle. So this is an exposed individual that is contaminated with a spore, but it does not have an infection yet. And within 2 days, the spore starts to germinate, forms a kind of an apresorium that produces enzymes and physical pressure to break through the cuticle and grows into the host to cause an internal infection. At this time, the spores that are here, they are so tightly attached that this individual is not anymore infectious for the other individuals. So we have a clear separation between an not yet sick but infectious individuals, and later on they are infected but not infectious. But then what happens is that there is a lot of healthcare and grooming going on in the exposed phase. So the nestmates try to remove these spores before they can actually integrate into the body. If this is not successful and an internal infection takes place, and then there is an immune response in the body, melanization, phenol oxidase activity is trying to hit this, and antimicrobial peptides try to make ruptures into the fungal cell wall. But if this immune response is also not able to clean the infection, then there is host death and fungal outgrowth. And this fungus is an obligate killing pathogen, so it requires host death to produce the next generation of fungal spores that you can see here, these green packets. And at this stage, a cadaver is then highly infectious. So we have 2 infectious phases: the exposure phase, where there is more or less distribution of the couple of spores in the colony. But then if you have a cadaver that produces 12 million spores at a time, and that would be really enough to cause 50% mortality in, at least in small groups of the colony. So what we typically do is that we then have, for example, our nestmate ants, and we have one treated individual, and we use in sham control, we just use a solution in which we otherwise have the spores suspended to apply it to this individual, because we know that the treatment is affecting also the behavior of the ants a lot. And the pathogen treatment is then containing these infectious particles. And then we observe mostly grooming behavior. And so grooming behavior means that you can have self-grooming or allogrooming. So the individuals themselves try to remove the contamination on their body by licking it off with their mouth parts. And the same happens in allogrooming. You here have an individual that tries to remove fungal pathogens from the body surface of its contaminated nestmate. And also there is a lot of chemical disinfection going on, not only mechanical removal of the spores, but also they apply their poison, which in these ants contains formic acid, which is a strong antimicrobially active compound that they really spray on these individuals, and they take it up into their mouth and directly apply it during grooming. So this is very effective. So what we do is that we have this experimental setup, and then what we create something like grooming networks. So and this means that we have then self-grooming is a circle around the individual, which is increased when you have a pathogen exposure, and also we can have directed networks, who grooms whom and at what intensity. So what we then observe is pathogen-induced behavior changes in the grooming behavior that we can visualize here as the change in grooming as compared to the time before like before exposure. So what we can see, obviously, is that the exposed individuals, they drastically increase self-grooming, whereas the nestmates, they don't seem to care. So it's not that they do a prophylactic upregulation of their own self-grooming. If you then look at allogrooming, what you can see here is that the nestmates groom somewhat more the exposed individual, but it's not always significant, or it's often not significant, but it's a trend. The nestmates among each other, they also don't groom more. But what is very clear is that the exposed individual itself is not grooming anybody else anymore. So, and this then leads to an allogrooming asymmetry that is very clear. So that means that the exposed individual is receiving much more allogrooming than it gives to the others. And this was something that was known already, but typically one had thought that this allogrooming asymmetry comes because the others just groom it much more. And we could show that it's mostly because the exposed individual stops its own allogrooming of nestmates. And we then tried to understand what would be the potential of these behavioral changes for disease transmission in the colony. So we asked our colleagues from the Helmholtz Center to develop an SIS model, infectious susceptible model. And we did some simulations and to predict the disease transmission in the colony based on these grooming rates. So what you can see here clearly is that if you increase your self-grooming rate, this is always very beneficial because the mean fraction of infectious ants in the colony will go down the more the exposed and infectious individuals are grooming themselves. And the same is actually true when this individual that is infectious just stays away from the others and does not get into contact where it could transmit the disease. So a decreased allogrooming of the exposed individual is also always beneficial. However, whether or not nestmates, healthy nestmates, should get into contact with an exposed individual and groom it or not, this is actually can go into both directions. And this is something that is very much dependent on the specific host pathogen system. So what is the probability of reducing an infection in the groomed individual, and what is the probability of the groomer to receive the disease? So there we cannot make a general statement, but it's really very much depending on the pathogen system. And in our host pathogen system, actually the thick line is always what applies to our system. Here you can see it would not even be predicted that the nestmates should allogroom them significantly more. So with our small-scale approach, we have been doing a lot of experimental work with this setup, and we also had the ants placed in a more natural environment with a brood chamber. And what we had observed there is that the directly exposed individuals, immediately after they receive this treatment, they change their spatial preferences. So these exposed ants leave the brood chamber. And our hypothesis, but we haven't really tested that yet, is that this is to then prevent transmission of the disease towards the brood, which is actually very susceptible to this fungus because the brood doesn't have such a strong cuticle yet. So it's very easy for the fungus to penetrate through this soft cuticle yet. Also, there is care behavior, the care behavior of the exposed individual. So an exposed individual stops any brood care and also the allogrooming. Whereas the nestmates, they don't significantly increase the care of the exposed individuals, but they increase the care towards the healthy brood. So they seem to like prophylactically also try to groom more this brood. And in the modeling, we could show that these behavioral changes are predicting a decreased disease spread in the colony. But so far, this was mostly really direct one-to-one interactions between the individuals. And what we are interested in is how are these interactions now scaling up to produce this social immunity at the colony level? And how is it changing really the interaction network at the whole colony level? So I'm interested to see how from one-on-one interactions this colony level disease defense is really emerging. And we have started a collaboration with the Keller group in Lausanne to really use their toolkit that I've shown you before, that we could observe whole colonies over a long period with the automated tracking. So what they do is that you can see here that they glue paper with barcodes basically on each individual ant. And these are so small because the ants themselves are something like 3 to 4 millimeters long. So what you see here is 1 by 1 millimeter papers that Laurent gets printed at the Swiss bank to get it so that no black basically is in the white, as if you would have a bad printer. So then they have built up a system where they then keep the humidity stable and everything, and where they can then do a real-time detection of the position and the orientation of all ants, and do this in 2 images per second. So they get huge amount of data on this. And this is how our setup looks like. So you have a nest area, and these individuals, they are already tracked. So each individual is then read out with its individual number. And the individuals can go to the foraging arena where they find water and sugar. So the first thing that you have to do, because you're never perfect in how you glue the tag on, the first thing you have to make a picture of each ant and to see in which orientation is your tag actually. And then you can you measure the ant as well, and you know, oops, this is always the thing. You have a trapezoid that you put around the ant, and with this you can then interfere. Basically, the only thing that you get is the information how all the tags are interacting with one another. But then you can infer how close are they? Is this within antennal contact reach or not? So this then gives you this social interaction network of the colony. But yeah, bear in mind that this is no behavioral detailed information. So our experimental design was very similar to before. We also had a solvent control, so a sham control, and a pathogen exposure, as we do in the small scale approach. Somehow this is always coming along, but yeah. So we can look at the baseline colony organization of these always 11 colonies with a queen and around 150 workers each. And then we looked at the colony for 4 days, and then we performed the treatment of 10% of the foraging workers, or 10% of the colony, and we took them from the foraging arena. And then we tracked for 24 hours and to see what are the pathogen-induced changes in the network. And so, yeah, what you can do is you really have the experimental data, and Natalie also then has been performing quite a lot of modeling based on this to see whether these changes would predict a decreased disease risk. So I will show you a few of our experimental data. So again, we found that the exposed ants changed their spatial preference. And if you look here at the time that this individual spends in the nest, you can see that it is much less time under pathogen exposure, so they go out more to the foraging arena. And they also change the type of interaction partners that they interact with. First I give you their interaction of the exposed individuals with other exposed ants or with other treated ants, and you can see here that pathogen-exposed individuals preferentially interact more with other pathogen-exposed individuals, and they thus also interact less with their healthy nestmates. So this means that there is some kind of clustering going on, that all the exposed ants cluster together and the healthy ones. And this also means that they have reduced types of overall interactions, and the time that is necessary for the exposed workers to enter in direct interaction with 50% of the nestmates. It's a bit of a complicated measure, but it's very much prolonged, as you can see here. So this means that there is some kind of, on one hand, a spatial change, but also a social isolation of these exposed workers. And this you can also look at when you look at network modularity, which measures the strength of division of a network into separate communities. And this modularity is also predicted to decrease the speed of disease transmission. And if you directly then measure the network modularity, you can see that it's really increasing in these pathogen-exposed colonies as compared to the sham-treated. So meaning that the interaction network becomes more heterogeneous, more substructured. And if you then look at the position of the exposed individuals themselves in this interaction network, we looked at the distance to the queen and the centrality of the workers. So the distance to the queen is just how many interaction partners would I need to reach the queen. And this is also drastically increased upon pathogen exposure, whereas the centrality, which means basically the connection that these workers have, is reduced. So it seems that all this would predict a lower transmission potential of the exposed individuals to their healthy nestmates. And so this means that we have a spatial and social isolation of the exposed individuals and a stronger substructuring of the network. And modeling is really has been performed, and I only give you one slide. But what she has been doing is that she simulated then the pathogen spread over these time-ordered networks that she could obtain, and performed a susceptible infected model without pathogen replication, and our preliminary data suggest really a lower disease risk in the pathogen experienced social interaction networks. So this all leads me to the kind of bringing together the small and the large scale approach. Means that there is really behavioral changes after exposure that are consistent between our small-scale behavioral approaches and the large-scale colony approaches, which for me is very important because that means that we can really use them complementary to understand the whole story. And both showing that there is a change in spatial use and also social interactions of the exposed ants. But the large-scale approach, in addition, can show that there are also network changes after exposure because of you have a higher substructuring and exposed ants becoming less central and more distant to the queen, which is the most valuable individual and where you want to not lead the disease to. So, yeah, as I said before, I think the two approaches can be valuably combined. And also consistently, epidemiological modeling predicts a reduced disease risk. But I'm an experimental biologist, and my feeling is always epidemiological modeling. If you twist around some parameters, you can maybe also find the opposite. So I think it is valuable, and I think it teaches us something, but it's still a prediction only. So what we really wanted to do is that we really observe the pathogen transmission in these colonies, and so we experimentally determine pathogen transmission. And we have developed methods for this to quantify the pathogen on each individual colony member after their interaction. So what we use is digital droplet PCR, which is a more sensitive way of a quantitative PCR method, where we target the DNA of the fungal pathogen, either the fungal pathogen gene, or we can also use their fluorescent labeling, GFP and RFP, that we target the fluorescent label. This digital droplet PCR means that you don't have a single reaction, but you split up your reactions into 20,000 small droplets in oil, where you can then see whether or not you have your target in there or not. And so we can go down to, I always say 5 spores, and my technician always says, yeah, say below 10 spores, because we have, but I think we can be very sensitive here, which is actually quite a low number when you compare the. Normally, an LD50 in the sense would be 30,000 spores that they pick up. So looking at 5 spores is really very sensitively showing us pathogen transmission to other individuals that are not even leading to a proper infection in these hosts. And the other benefit of this method is that you have an absolute quantification. You don't need the standards with it, but you really measure directly whether this is there or not. And so here we can study simultaneously the pathogen removal from the exposed individual. We know what we put on, what they had at the start. So they had a given number and the others all had 0. And then we can see how much did they decrease and how much did the others increase, the caregiving individuals. And what we have developed now is the method to target we have the same fungal strain in RFP and GFP labeled, and we target with our PCR the RFP versus the GFP, so we can also have 2 individuals that are exposed in the colony, also with different amounts of spores, if we like, or that are of different roles in the network, and measure simultaneously the disease spread or the pathogen spread from these 2 individuals. So the experiment that we are currently analyzing in my lab is the PhD thesis of Barbara Casillas. And so again, we have the similar setup. It's a small-scale approach. It's only 6 ants. 2 of them are treated. And we observe them before and after the treatment. And here what we do is really a video analysis of individually color-coded individuals. So this is a lot of video analysis that we are doing here, but we wanted to have all the exact grooming data here, because if you only look at interactions, you cannot disentangle whether the ants are just close, whether they are just antenating, smelling who is there, or whether they really groom off and pick off the fungal spores. And in this sham control treatment, then after the first period before treatment, where we get to know our networks, we treat both individuals with a sham control. And in the pathogen exposure, you can then have both individuals treated with different colored fungus, and you can also have all combinations in between of one sham treated and one red or green. So from this grooming network, we can then get a grooming matrix, so to speak, where we look at the receiving ant and the performing ant, and this is like a heat map information. So our data indicate that before treatment, there's relatively equal balance of allogrooming among the individuals, and after treatment, and this is only sham treatment of the individuals, you can already see that the 2 directly exposed ants, they have a very low allogrooming, so they keep as before, more or less, whereas they are groomed by the others more. So you can already see baseline changes just because you somehow treat these individuals, they are reacting to this. So that is for us very important, that we always compare our changes to the sham control and not to an untreated control individual. If you have one sham treated and one pathogen-treated individual, you can see, again, both treated individuals don't increase their grooming, but they are groomed more. And if you like, you can see that the pathogen one is probably groomed more, but we haven't done the statistics on this yet. If they are both treated with a pathogen, then you can see an even clearer effect here. So again, the pathogen-exposed ants are ceasing the grooming, but there is an increase that is significant or not. We haven't tested it yet of the nestmates. So we want to know what's the effect on the pathogen spread. Then I think one has to make a very important distinction between pathogen spread and disease spread. Because with sanitary care, we know that pathogens are transferred from one individual to the next because the groomer will receive the spores that it picks off from the other one. However, this does not necessarily mean that the grooming individual will develop a disease because of that. Because what they do is that they take up these infectious particles into their mouth and they have a mouth pouch, which is the infrabuccal pocket. And we have talked about this yesterday, that in these infrabuccal pockets, what they will do is that they actually disinfect the pathogens that they take up by labial gland compounds, but also by their own poison, their formic acid-rich poison that they take up into their mouth, and that is reducing germination to at least 50%. So this means that if we look at fungal DNA with our PCR, we pick it up, but we don't know whether they are actually germinating or not. And also, they will be spit out after some time, and they will not be leading to disease, very likely. So, yeah, I've brought a quick video that shows you here an ant that is spraying an exposed pupil with its formic acid poison. And you may see, I'm not sure whether the light is good to see it, that there is some kind of color change from blue to red when this acid enters. This is how we can quantify this formic acid spread. And now what the ant is doing, it is taking out the infrabuccal pellet that it has produced, and it's going to do that in a second or so. Now it comes out of the mouth, it's taken up, and it will be placed somewhere in the nest, if you can follow it. And this is all compacted fungal spores. So that means that if we collect an ant and it has its mouth full of pathogens, we will have this detected, but it's not a disease-relevant pathogen. So I would like to make this distinction between, say, controlled and uncontrolled contamination. So the controlled pathogen transfer to specific disease defense organs, the infrabuccal pockets, where they're disinfected and then discharged, we expect high amounts of particles that are either still infectious or already disinfected, but we expect them to be related to very little disease risk, actually. Whereas on the other hand, you can have uncontrolled pathogen transfer by, say, accidental contamination, by rubbing off, by walking past an individual and getting it on the back or something. So where we can actually have quite low amounts of infectious particles that provide a high infection risk, and where also the detection of the ants, because it's somewhat unnoticed by the ant itself, so maybe they are not cleaning it so clear as if they know that they have it in their mouth. Also, this particular pathogen is very highly dose dependent. So only a high, I would say a high infection load is causing disease, and a low infection load is actually non-disease, doesn't show any disease symptoms, but it actually induces some immune stimulation, and even then is some kind of
vaccination, if you like, or variolation in the closer sense, a protective effect, which causes a protective effect upon secondary exposure. And there is some correlation between the exposure dosage that an individual gets and the later received infection likelihood, whether they have a high or low level infection. Sometimes, I mean, a few spores can already lead to a high infection, but in general, the more you are exposed to, the more likely it is that you will develop a disease. And tomorrow I will actually talk more about the social immunization, about these spread of low-level infections in the helpers when they have groomed off other individuals. But today I would just like to show you our preliminary data. So what we do to disentangle between these, I would say, disease-relevant pathogen transfer and not, is that we cut the end in half. Basically, we make one PCR of the head and we make one of the remaining part of the bodies. And our preliminary data show that there is actually, we can detect very high pathogen loads in the head and much fewer of this uncontrolled contamination on the body. And that what we can detect is really mirroring the behavior that we can see. So, for example, this individual has groomed a lot the red end and only little the green end. And we can see a much higher representation of red spores in the infrabuccal pellets than of the green spores. And here the opposite is true. You can see more green than red after having had very much allogrooming of the green individual. This is now not yet time resolved, but we can also get this picture. We know, oh, it has groomed the green end for 5 seconds, and then it made some self-grooming, and then it went to 2 seconds to the red end. So we're still in the middle of this, but I wanted to show it to you that I think we have a quite good toolkit at hand with which we can really look at this. So if we come back to the small and large scale approach, I think we can look at the side of the host and see that there's individual and pairwise behavior is changing upon pathogen exposure. And the small and large scale approaches need to be combined actually to get the full picture. And we also need to get this interaction network data in addition. The pathogen actually, for the pathogen, it's important to know the host interaction network because the pathogen can travel along this network. It's a theoretical pathogen transmission network that we can also use via modeling. But with our experimental approach, what we can do in addition is to really look at the realized pathogen transmission along this network. So I try to make a step forward from the pathogen also towards the disease, to not only look at pathogen transfer, but also how it then relates to later disease outbreak, and from short-term transmission to long-term colony fitness. Because you can see here that, for example, if we have an initial infection in the colony, what we can then start looking at is how is the pathogen transmitting. And if we perform longer term studies, we can then see which of these individuals really get sick. And in this case, this would mean that they would die off. Whereas these individuals, and here they are not represented in the next time in the network anymore, whereas the other individuals that have seen the pathogen but survived it, they actually become immune. And then the host can also recover and produce new individuals to make up for the loss of workers. So it can tolerate the disease by making new individuals, whereas the loss of the disease of the queen could not simply be recovered. So here we think that we need for example, resistance mechanisms to really protect the queen, so to reduce the pathogen load in the colony and also make sure that the queen is not dying. But on the other hand, maybe you don't have to really be so careful to prevent infection of all your worker force. Maybe sometimes it's a better strategy to not invest too much into individual resistance of each worker, which is very costly, but instead then reduce the harm caused by the infection and be able to regenerate the worker force very much. So I'm now trying to see what is the resistance and what is the tolerance mechanisms at the colony level. So I think to bring me back to the sociality and disease picture, the exposed individuals, as we have seen, are the ones that change the network a lot because they do spatial and social isolation, and they also try to eliminate the pathogen by self-grooming. But they are the ones that kind of retract themselves from the social network and reduce the risk of transmission. Whereas I think the nestmates, they perform sanitary care and disinfection. They perform it sometimes at the level of the other individual and others more to protect the whole colony. But mostly what they do is that they eliminate the pathogen. And for example, these can maybe not have so strong effects on the host behavioral interaction networks because some of these behaviors are very fast and very rare behaviors. For example, spraying the formic acid on somebody. It only appears once every 4 hours, but it has a very strong effect in reducing the pathogen load or the germination ability, but it doesn't change too much the interaction network. So that's why I think we really have to study both the detailed behavior and the network and the pathogen spread. So this is what we do in ants. So can we generalize from this? Can we make any conclusions? And also I want to ask the question, are social insects good model systems for disease in societies in general? And of course, my answer would be yes, at least part of it, because I think that social insects, they can be used as a very good experimental system, as a high throughput system where you can have high statistic power. They are societies in a petri dish, right? So you can have multiple replicates and you can have complete surveillance of every little step that every ant is doing. They live in this two-dimensional environment that we can completely track and monitor them. And we can even try to manipulate the interactions, and that is my next step, where I want to prevent them from leaving the brood chamber and see whether this is then really increasing the disease risk of the brood or something like that. Whereas in humans and primates, you often only can do retrospective analysis and more correlative evidence, whereas I think social insects can be used really to manipulate. But then I also have to say that I don't think not that every aspect of disease in society ants are a very good model system, because they are very different from other societies because they're actually superorganisms. So it's not that every society member is totipotent and will introduce it or have its own reproductive potential, but they act really together similar to a body. It's like an organismal immunity that we find in these superorganisms. This makes it easy to then look at the colony fitness effect of these society interaction network outcomes, but I think it makes it it doesn't allow for a complete parallel setup in the ant societies and in human societies. So with this, I would like to thank my team, the social immunity team at IST, and I would like to promote that we have a graduate school and postdoctoral funding opportunities at IST, and we're always interested to have new people come in, if you like. So thanks a lot for your attention.
Questions, and we will need to use the microphone so people who are watching from afar can understand our questions. Only time for a few today.
I was just wondering what control mechanisms or solutions you put on the ants that would be sufficient to mimic an infection.
So you mean that one takes a non-pathogenic proxy?
Right.
So we have been playing around with some fungal spores that are actually you can irradiate the fungal spores, and you can also try to make them smell less. So this is one control that I would really like to introduce, that you can take also a non-pathogenic but very similar to a proper pathogen, that you can then look at the spread of this when they don't show the induced behavioral component, because they don't realize that a pathogen comes in. What other people have been doing is that they used beads, for example, fluorescent beads that they give to bees that are then exchanged via regurgitated food. Because here I was talking about an externally infecting pathogen, but you can also have a lot of orally infecting pathogens, and they are typically transmitted by food regurgitation networks. So here we have a grooming network, but you can also have this trophallaxis network, how it's called actually.
So when you did the network analysis, and one of the conclusions was there is the modularity increased, with the narrative being that that should isolate off the infected individuals. But before that, you said that the allogrooming asymmetry was actually set up by individuals just backing off their own allogrooming when they got infected. Do you think, if it is true that the modularity you see in the networks is really isolating these individuals, don't you think those two effects could end up kind of balancing out? So they end up not allogrooming that much, plus they back out from those who would groom them. So, I mean, how do you sort of reconcile both needing isolation and also needing to be groomed?
Mm-hmm. Yeah, so I think one question that we still have not really answered is whether this isolation, who is the active part in this isolation? So the idea is that they will we don't see any aggression by nestmates. So our feeling is that they really self-retract. And there's even studies that suggest that individuals close before death are actually leaving the colony completely. So the idea is that they do altruistic self-removal from the network. Yes, I think it is. I was also surprised myself that there is not a significant value of increased allogrooming to the exposed individuals. You can see that it goes up, but it's actually not so strong as everybody has expected. And that, I think, is really because they are somehow isolating themselves. But yeah, it's a tricky issue, and I don't know where the individuals or where each colony finds the balance will probably really depend exactly on the particular pathogen system and the dosage that you apply. So in this experiment that I've shown you with the red and green ants, so what we actually are interested in this is that we apply the same number of spores in the colony. So if we would apply it only to a single ant, or if we apply it equally balanced to two ants. So we have 50% or 100%, and we want to know, is this affecting the interaction network as well? But I don't have an answer yet.
Thank you so much, Cynthia. We really appreciate it.
Thank you.